Orbital Gardens, this is Mission Control. We are confirming acquisition of your signal. You are live in 5, 4, 3, 2.
Hello and welcome to Gardeners of the Galaxy, the podcast for all of the sentient beings in the universe who have a passion for plants. I am Emma the Space Gardener and I will be your host as we explore gardening on Earth and beyond. Wisconsin fast plants are having a bit of a moment. We've watched them take to the skies on 2 Blue Origin suborbital flights, with Emily Calandrelli in November 2024 and Amanda Wien in April 2025. They are also being used in astrobotany experiments by researchers at the United States Air Force Academy. Fast plants are a type of Brassica rapa specially bred for use in experiments. Among other things, they go through their entire life cycle, from seed to seed, really quickly, hence the name. They're used a lot in classrooms here on Earth, and there's a special variety called astroplants, bred for space experiments. In 1997, astroplants became the first plants to grow from seed and produce viable seeds in space, which were then used to grow a second generation of space plants. That was on the Mir space station, and the investigation almost came to a crashing halt. You can hear more about that in episode 38, of Nominal Spaceplant Experiments, my blooper reel, if you like, of experiments that didn't go according to plan. Today I'd like to tell a different story from the Astroplants archive, which starts with a cosmonaut born in Ukraine. Leonid Kadenyuk, a pilot and an engineer, was accepted for cosmonaut training in 1976. As well as his general spaceflight training, he studied botany and biology, learning how to perform scientific research. When Ukraine declared independence from the Soviet Union in 1991, Kadenyuk retained his Russian citizenship in the hope that he would eventually fly to space. But seeing that Ukrainian cosmonauts had been trained for but never given a space mission, Ukraine's first president, Leonid Kadenyuk, and I apologise if I'm not pronouncing these names correctly, negotiated a deal with the US and NASA. A Ukrainian astronaut would fly on the Space Shuttle and conduct spaceplant experiments, since botany and agriculture are such an important part of life in Ukraine, which is known as the breadbasket of Europe. The project was called the Collaborative Ukrainian Experiment, or Q, and it led to Kadenyuk renouncing his Russian citizenship, returning home to Ukraine, and becoming a researcher at the Institute of Botany in Kyiv. By 1996, he was at Johnson Space Center, working with astrobotanist Mary Musgrave, who led the team that conducted those astroplants experiments on Mir. The science component of Q was a collection of plant experiments involving astroplants, soybeans, and moss, but the scope of the mission expanded to include an educational component, with students in the US and Ukraine duplicating some of the space experiments on Earth. It was given the unmanageable name Collaborative Ukrainian Experiment, Teachers and Students Investigating Plants in Space, for which the acronym is QTIPS. It was Space Shuttle Columbia that finally gave Kadenyuk his ride to space in November 1997. He was a payload specialist on STS-87 alongside 4 NASA astronauts, a Japanese astronaut and a Russian cosmonaut. At the time, he said he had been waiting for this opportunity for 36 years, ever since he was 10 years old. Over the course of the next 2 weeks, Kadenyuk grew plants from seed and tended to others that had been grown before the flight. He pollinated them using a bee stick, a dead bee glued to a toothpick, a feat that involved wearing big magnifying goggles. Using a bee stick was a relatively new technique that Michael Foale had used during his successful experiment on Mir. Several of the investigations used the Plant Growth Facility, PGF, which was taking its first flight. The PGF was an updated version of the Plant Growth Unit, PGU, that first flew on STS-3 in 1982 and was designed to study seedling growth and lignification, the formation of wood. One PGU fit into a mid-deck locker on the shuttle and held 6 plant growth chambers, PGCs, and systems supporting plant growth. These included fans and a heater to regulate temperature, and 3 fluorescent lamps controlled by a timer. The PGU was used for experiments over 15 years. The updated PGF was the same size, but with much better lighting, an ethylene filter, and an air management system capable of controlling humidity, carbon dioxide, and temperature. The brick hardware, that's biological research in canisters, was also used for Q, and that basically involves growing things in petri dishes. Okay, so I won't go into massive details about each of the experiments, but I'll give you an overview and I'll put links to more information in the show notes for you. They were divided into two groups, the primary experiments and the secondary experiments. The primary experiments were BPAC, Brassica rapa photosynthetic apparatus in chamber, B-STIC, Brassica rapa seed terminal growth in chamber, SoyMet, soybean metabolism, SoyPat, soybean pathogen interactions, and SPM, Space Moss experiment, parts A and B. The two astroplants experiments explored how spaceflight affected the photosynthetic apparatus of seedlings at different stages of growth, pollination, and fertilization. The two soybean experiments explored the interaction of microgravity and ethylene on growth and metabolism and host pathogen interactions using a soil-borne pathogen that causes stem and root rots. And the Space Moss experiment looked into how microgravity affects cell differentiation and responses to red light using two different types of moss. Moving on to the secondary experiments, they were amino, genetics, lipid, phyto, and roots. They all used astroplants. Amino investigated spaceflight effects on amino acid content. Lipid had a similarly self-explanatory name. It looked at spaceflight effects on lipids and fatty acids, while phyto was about plant hormones, and roots explored gravity effects on the structure, function, and organisation of root cells. Genetics also used soybean tissue to determine the expression of metabolic and stress-related proteins and RNAs in the spaceflight environment. Early inspection of the plants grown on orbit showed the result of some higher than anticipated temperatures, but in the chambers where the temperatures were closer to optimal, the plants appeared normal. And while Kadenyuk conducted his space experiments, the student researchers on the ground replicated his every move with their own plants. During a video downlink, Kadenyuk spoke to students in Kyiv and the US, answering questions about similarities and differences in the way the plants grow on orbit and on the ground. Questions ranged from whether the plants and their blossoms remain the same on orbit as on Earth, to whether Kadenyuk was having the same trouble keeping his bee attached to the stick. One student noted that they were finding a lot of variations in their astroplants, asking whether Kadenyuk was also seeing this on his space plants. He replied that the space-flown plants were selected from a large number of astroplants to be as similar as possible to minimise the variability of the plants in orbit. He also confirmed that the flower buds in space developed the same as on Earth, and that NASA used superglue to make the bee sticks, with the best ones being selected for flight. At a wrap-up symposium held later at Kennedy Space Center, about 20 teachers gathered to share the results of the Earth control experiments. They had found that the students had really been excited to participate in a NASA experiment. The results varied though. Some teachers said that the plants were the worst they'd ever grown. For others, they did quite well. Kadenyuk's time in space ended on the 5th of December when Columbia touched down safely at Kennedy Space Center. He would later comment on how he saw the mood of his colleagues improved as they smiled, floating close to his plants, concluding that plants were important on spacecraft to maintain the psychological balance of cosmonauts and astronauts. After the flight, Kadenyuk spent much of December 1997 touring American cities before returning to Ukraine in January 1998 to be greeted as a national hero. He would later become a member of parliament in Ukraine and a science advisor to the president and the State Space Agency of Ukraine. He wrote a book, only available in Ukrainian, about his spaceflight, took part in international conferences and made a point of continuing to work with young people. He died in 2018 at the age of 67. So fast plants have played a significant role in astrobotany history and are due to take a giant leap in the near future. They're being included in the Lunar Effects on Agricultural Flora LEAF project that will head to the moon on the Artemis 3 mission. It's an international effort led by Spacelab Technologies and involving researchers from the Plants for Space Collaboration, NASA Kennedy Space Center, Purdue University, the University of Colorado Boulder, and the United States Department of Agriculture. Red and green fast plants will be growing alongside duckweed and Arabidopsis in a plant growth chamber on the lunar surface. So that's definitely something to look forward to. In the meantime, I'll add links to the show notes if you want to learn more about Leonid Kadenyuk and Q, and you'll find those at spacebotany.uk. That's it for this episode, so I will hand you back to Mission Control. Capcom, can you let the technicians know they've really nailed it with the latest zucchini experiment? The new plants are so productive I've been able to send several batches of fresh squash to the moon colony.
Orbital Guidance, this is Mission Control, confirming termination of your signal. We've had a request that you stop supplying your surplus zucchini to the moon colony. They've had about as much zucchini bread as they can stomach. Mission Control out.